Hello, everybody. In this lecture, we're going to talk about the process of translation. So let's get right into it. By the end of this module, you should be able to explain the process of translation and compare and contrast standard genetically encoded amino acids to non-standard genetically encoded amino acids. So to start off with translation, we must have the ribosome. The ribosome is a protein that builds proteins, essentially. Ribosomes have both a large and small subunit. They are inactive by themselves, but when you put them together, you get um, your complete ribosome. So here we have a eukaryotic ribosome, which is the 60S large subunit, the 40S small subunit, which makes up our 80S ribosome. This is different from our prokaryotic ribosome, which is a 50S large and a 30S small to make a 70S complete ribosome. So the genetic code has your four nucleotides. Those are going to be A, U, G, C in RNA. And the RNA ones are the ones that are important because when we're thinking about translation, we are translating the mRNA to make a protein. So three nucleotides in a row are going to make up a codon. And therefore, there are 64 unique codons. That's the triplet code of four, possibilities times four possibilities times four possibilities. 4 to the third power is equal to 64. So of your 64 codons, there are 60 what we call sense codons. They code 22 different amino acids. There are 3 nonsense codons, which are stop signals that do not add amino acids. And then there is 1 start signal, which generally adds an amino acid. So, and those codons are the way that we determine the production of all proteins. And so here is how the genetic code works for your 20 standard amino acids and the stop codons. So what you see here is a organization of the first base in the codon in green, the second base in orange, and the third base in this dark blue. And we see here, if I were to say, what does UCC code for? You would come over to U, C, C, and find that that codes for serine. I think the thing that you notice right off the bat is that with 60 possibilities and only 22 amino acids, or in this case, 20 amino acids, what we're talking about here is a lot of redundancy. So you can see that UCC makes serine, so does UCA, UCG, UCU. So any alteration in that third amino acid is still going to give you serine as long as the first two, or sorry, that third base is still going to give you serine as long as the first two are UC. And so you see this across the board that there's some level of um, of built-in redundancy, with the exception of the start codon, which is methionine, which is always AUG. And so this is what we're talking about. A single amino acid can be coded by multiple codons. The biggest example here where you have the most possible differences is for leucine. So leucine has all of these variabilities: UUA, UUG, and then all four CU possibilities. And this is something that's going to be there to help protect against genetic changes. And what that means is that if you get a single base pair difference where this U gets mutated and becomes a C for some reason, or if the C is mutated and becomes a U, whatever it is, you're going to still get a leucine. It doesn't matter. And so that's why you have this kind of built-in redundancy. When you have problems, especially in that third base pair, or that third base in the codon, then it doesn't actually affect which amino acid is added to your protein. And so the process of performing translation is very fast. For example, E. coli can add up to 20 amino acids per second, and it only misreads about 1 in every 10,000 codons, right? So that's most proteins don't have 10,000 amino acids in them. Just like in our other two processes, the three steps are initiation, elongation, and termination. And so these are the main players. Um, you've got your ribosome. It's got three sites: an E site, a P site, and an A site. E is your exit, P is your peptidyl site, and A is your acceptor site. You have your tRNA, which has an anticodon and it has an amino acid. And then you have your mRNA, which is broken up into a series of codons, which are these triplet codes, these triplet bases. Those codons are going to bind to the anticodon of the tRNA. Now, I like to kind of come up with a little mnemonic for remembering the sites in the ribosome. The acceptor site or the A site, I like to think of it as the arrival site, and the P site is the party site, and the E site is still the exit site, right? And so I like to say you go ape at the ribosome party. So you arrive at the party in the A site, you party at the party in the P site, and you exit the party in the E site. So here we are. We've got to start the party though, right? And so the person who starts the party in this case is going to be methionine. And so AUG, when the ribosome finds that start codon, AUG, it's going to bring in the initiator tRNA. That initiator tRNA is going to have the methionine on it, and its anticodon is going to pair with the codon of the mRNA. All right. And so this is the only one that enters at the P site because it's starting the protein party. All right. So we see over here the complementary base pairing between the anticodon on your tRNA and the codon on your mRNA. And in prokaryotes, the translation is going to be initiated even before transcription concludes. And so multiple ribosomes latch onto that single mRNA molecule, forming what's called a polysome. And so this can just happen very rapidly. This is different than in eukaryotes, right? Because eukaryotes have to actually take the transcript out of the nucleus. So once we've initiated replication, so in initiation, the ribosome has to form around your mRNA. It moves along that mRNA until it finds the start codon, which is going to be AUG, which is going to code for methionine. Then what's going to happen is that new tRNAs are going to be added into the A site, right? They're going to arrive at the protein party at the A site. And so the anticodon on the tRNA is going to match with the codon on the mRNA. And then the ribosome is going to move in this direction, all right? And as it moves in this direction, which is 5 to 3, we're always moving 5 to 3, the methionine on this old tRNA is going to be added onto here. Now, this picture is actually bad. This tRNA down here should ultimately be popped off. Or the amino acid should be popped off of this tRNA. So I'm going to go into the slideshow after this and edit that picture. But you do see that the methionine is on here. And so if we look at the way that this is set up, when this guy starts the party in the P site, all the new party members are going to come into the A site. All right. So that tRNA is going to arrive at the A site. And then as the ribosome moves in the 5 to 3 direction, the methionine is going to be transferred from this tRNA to this tRNA. And then you're going to get a growing polypeptide chain on the tRNA that's in the P site. And whichever tRNA is in the E site gets bumped out. It exits the party. And so it's going to keep doing that over and over again. And each new Um, uh, each new tRNA is going to bring in an amino acid based on the anticodon codon pairing. And then ultimately, you're going to get to a point to where you're going to get to a stop codon. And the stop codon, you're going to terminate the actual process uh, of translation. And so to review, if you want to go to the ribosome party or the protein party, you arrive at the A site. Okay, you arrive to the party of the A site, you party at the party of the B site, you exit the party in the E site. So what that means is that you bring in your tRNA here. When you get to the P site, you're going to hold on to the entire protein, right? So it's going to be added to your end. So this is what this picture here is showing. And then when you get to the E site, it's time for you to leave. You've transferred the party to a new tRNA. So we're going to briefly talk about post-translational modifications here, and then we'll talk a little bit about some other gene regulation things in another lecture. So for a gene to function, there's usually going to be some sort of post-translational modification, such as trimming, or adding on additional organic factors such as sugars and lipids, or inorganic factors such as metal ions and phosphate. And so what this means here is that in trimming, what you're doing is you're going to come in and actually cut off bits of the protein sequence. So sometimes, especially common one would be cutting off that methionine, right? So that start codon is necessary to get the process of translation started, but not all proteins actually have that methionine in its functional form. So what they'll do is come in and trim off the methionine, and then you will get, the rest of the protein chain, for example. Another example would be something like when you add on to, when you're finished making this, you can add on like a sugar to make some glycoproteins and those sorts of things, or glycopeptides. And when you add those sugars on, they help signal to where they need to go and can also be involved in their functionality. There are a couple of non-standard genetically encoded amino acids. So we have our 20 standard amino acids that we use for making our proteins, and then we have these two non-standard ones. So under these specific instances with stop codons, you have UGA and UAG, can actually cause the addition of selenocysteine or lysine. And so for these situations, I'm not super worried about you having a detailed understanding of that situation, other than just being aware that these things can exist. All right. So the big thing I want you to pull out of this is the ability to explain the process of translation. I want you to also to be able to compare and contrast those standard and non-standard amino acids. But the real thing is kind of understanding how the standard amino acids are chosen, and then just being aware that those non-standard amino acids exist and they can be coded by stop codons in special circumstances. All right, with that, I hope you enjoyed, and I'll see you soon.